Welcome to Dr. Mercola's Cellular Wisdom. Stay informed with quick, easy-to-listen summaries of our latest articles, perfect for when you're on the go. No reading required. Subscribe for free at mercola.com for the latest health insights.
Hello, and welcome to Dr. Mercola's Cellular Wisdom. I'm Ethan Foster, and today we're exploring how the microbes in your gut can shape your resilience to stress and influence mental well-being.
I'm Alara Skye, glad to be here. We'll examine new research on the brain-gut microbiome system, why a balanced microbiome matters for mood, and practical steps you can take to support beneficial bacteria.
Let's start with what scientists call the brain-gut microbiome, or BGM, network. Recent work in Nature Mental Health shows that the trillions of organisms in your intestines exchange signals with your brain and nervous system, directly affecting how you handle pressure.
The study compared people who bounce back quickly after challenges with those who struggle. Highly resilient participants had distinct gut communities that were more active in energy production, environmental adaptation, and inflammation control.
On brain scans, those same individuals showed stronger connections in reward circuits and more efficient regulatory pathways, preventing an exaggerated stress response.
Importantly, the activity of gut bacteria predicted resilience better than brain imaging alone. That tells us the microbial environment is not a side note. It is central to mental health outcomes.
When that environment is disturbed, a state called dysbiosis sets in. Evidence links dysbiosis to anxiety, depression, bipolar disorder, and even neurodevelopmental conditions such as autism spectrum disorder.
One marker is a drop in butyrate-producing bacteria. Butyrate is a short-chain fatty acid that nourishes colon cells, maintains the gut barrier, and modulates inflammation. Lower butyrate often correlates with higher depressive symptoms.
That barrier function matters because a leaky gut allows toxins and undigested particles to enter the bloodstream, driving systemic and neural inflammation that can amplify mood disorders.
Oxygen-intolerant bacteria, such as Akkermansia, create butyrate and other SCFAs in an anaerobic, low-oxygen colon. Modern diets rich in seed oils and other mitochondrial stressors allow oxygen to creep in, favoring harmful oxygen-tolerant microbes that release more endotoxin.
More endotoxin means more inflammatory signaling and a harder climb back to equilibrium after stress. Repairing gut ecology is therefore a logical target for improving psychological resilience.
Practical measures begin with reducing linoleic acid, the dominant fat in seed oils. Aim for under 5 grams daily, or 2 grams if possible. Removing those oils for at least 6 months gives mitochondria room to recover.
Next, modulate carbohydrate sources. If digestion is compromised, start with easily absorbed options such as diluted dextrose or small amounts of sucrose water, then progress to white rice and whole fruits.
As tolerance improves, introduce complex carbohydrates gradually. Root vegetables, custom pastas, and finally legumes and whole grains, while monitoring for gas, bloating, or discomfort that signal excess endotoxin production.
Throughout this progression, prioritize fresh, unprocessed foods. Whole fruits supply polyphenols that friendly bacteria convert into beneficial compounds, while fiber feeds SCFA producers once the gut barrier is stable.
When conditions are right, Akkermansia supplementation can strengthen mucin production along the intestinal wall, tightening junctions and lowering permeability.
Choose a capsule with delayed release technology and take it on an empty stomach, so live organisms survive the 2-hour journey to the colon rather than dissolving in the oxygen-rich upper tract.
Dosage in human trials ranges from 100 million to 10 billion colony-forming units per day. Metabolic studies, such as those on insulin sensitivity, tend to use the higher end, around 10 billion CFU.
Lower doses, about 1 billion CFU, still appear effective for fortifying the gut barrier and easing liver stress. The optimal amount may depend on individual microbiome composition.
Remember, potency numbers can be confusing. CFU counts viable microbes. AFU measures enzyme activity, live or dead. TFU includes every cell, even those that cannot function. For probiotics, CFU remains the benchmark.
Pasteurized or heat-killed, Akkermansia products often advertise high TFU values, but dead cells cannot colonize or produce metabolites. Living bacteria are required for sustained benefits.
None of this will succeed if mitochondrial output stays low. Energy-starved colon cells cannot maintain the anaerobic environment that Akkermansia demands, so addressing lifestyle toxins is foundational.
Key mitochondrial supports include eliminating excess seed oils, minimizing exposure to endocrine-disrupting chemicals, and reducing non-native electromagnetic field stress where feasible.
Adequate sleep, sunlight, regular movement, and nutrient-dense foods such as magnesium-rich leafy greens round out a program to restore cellular energy and, by extension, microbial balance.
Large-scale analyses in translational psychiatry underscore this link. Volunteers with lower microbial diversity consistently displayed higher cortisol awakening responses, highlighting microbiome influence on endocrine stress pathways.
Cortisol is not inherently harmful, but chronic elevation disturbs hippocampal function and sleep architecture. Rebalancing gut flora moderates the spike, leading to more stable circadian rhythms.
Another emerging theme is early life programming. Animal models show that antibiotic disruption in infancy changes social behavior and anxiety scores months later. Effects reversible with fecal microbiota transplant.
Translating that to humans suggests pregnancy and early childhood diets rich in whole foods and minimal seed oils may lay a durable foundation for stress adaptability later.
Let's touch on cooking methods. Boiling potatoes reduces oxalates, while cooling and reheating rice increases resistant starch, feeding butyrate producers without spiking post-meal glucose.
Similar principles apply to beans. Long soaking and pressure cooking decrease lectins, improving tolerance as your microbiome transitions toward a balanced, low endotoxin state.
Regarding supplements, synergy matters. Trials combining Akkermansia with inulin-type fructans report enhanced colonization and improved insulin sensitivity beyond either component alone.
Prebiotics act as selective fertilizers. Introducing them slowly, starting at 2 grams daily, prevents excessive gas while encouraging beneficial species to outcompete opportunists.
Monitoring progress can be as simple as tracking mood and digestion in a journal, then correlating trends with dietary changes to personalize your approach.
For quantitative feedback, stool sequencing kits now measure Akkermansia abundance and butyrate gene counts, offering a functional snapshot instead of purely taxonomic labels.
Importantly, resilience extends to immune regulation. Balanced microbes dampen excessive cytokine release, helping the body reset after acute infections or intense exercise.
That same anti-inflammatory signaling protects the blood-brain barrier, reducing neuroinflammation and preserving cognitive clarity during periods of high mental demand.
Clinical researchers in Belgium gave overweight volunteers 10 billion CFU of live Akkermansia daily for 3 months and recorded a 3-pound fat loss without any prescribed diet change.
Participants also showed a meaningful drop in total cholesterol and improved insulin sensitivity, reinforcing the idea that gut interventions can modulate systemic metabolism as well as mood.
Separate trials using 1 billion CFU in cirrhosis, patients strengthened gut barrier proteins and reduced serum endotoxin, outcomes linked to lower cognitive fog known as hepatic encephalopathy.
These real-world results align with mechanistic studies showing that Akkermansia increases mucin thickness, limits pathogen access to epithelial cells, and lowers upstream inflammatory signaling.
Beyond supplements, everyday habits influence colon oxygen levels. Prolonged sitting compresses abdominal blood flow. Regular walking encourages peristalsis, disperses gas, and maintains the low oxygen niche beneficial microbes need.
Cold exposure and diaphragmatic breathing similarly enhance mitochondrial function, boosting the NAD ratio that supports cellular respiration and, indirectly, the anaerobic gut environment.
People often ask about fermented foods. While sauerkraut and kefir introduce many species, their CFU counts are inconsistent. They complement but do not replace targeted clinical-grade Akkermansia.
Still, traditional ferments provide organic acids and enzymes that precondition digestion, making it easier for supplemental strains to establish. Introduce them cautiously if histamine reactions are an issue.
Another metric worth tracking is stool pH. A slightly acidic range, about 6.0 to 6.8, signals adequate SCFA production, whereas alkaline values may indicate dysbiosis or low fiber intake.
Pair pH checks with periodic blood markers, such as fasting insulin, high-sensitivity C-reactive protein, and gamma-glutamyl transferase, to monitor systemic responses to microbiome-focused lifestyle changes.
We've covered only one aspect of the brain-gut relationship. Other factors such as vagus nerve tone, breathing patterns, and social connection also interact with microbial signals, and we'll explore those in future episodes.
For now, focus on what is within immediate control: food quality, environmental exposures, and supplement timing. These leverage points are simple, but their physiological ripple effects are profound.
That wraps our discussion. Thank you for joining us on Dr. Mercola's Cellular Wisdom.
Stay tuned for more evidence-based conversations that put you in charge of your health. Until next time, take care.
Thanks for watching. Subscribe now and click the notification bell so you never miss an update. See you in the next video.